Welcome to Prospective Doctors MCAT Podcast, brought to you by Med School Coach. Each week, Sam breaks down the highest yield MCAT topics so you can score as high as possible on test day.
Hello and welcome to Prospective Doctors MCAT Basics with your host Sam Smith. The goal of this podcast is to cover the highest yield topics on the MCAT and provide you with some sort of insight into the questions that the MCAT really likes to ask. This podcast is going to cover the immune system, and the immune system really breaks down into two main categories. That is the adaptive immune system and the innate immune system. And originally I was going to do this podcast on both those systems, but it was just going to take up too much time. So I'm splitting that up, and therefore this podcast is going to be on the innate immune system. I'm going to start by introducing the cells of the innate immune system, and then I'll go through kind of the different layers of the innate immune system, break each of those down. I'll spend quite a bit of time discussing the inflammatory response. And this material is going to show up in one out of the four sections mostly, which is going to be the biochem section, where you'll see a lot of these different physiological systems. And like a lot of these different podcasts, this is kind of a surface level view of the innate immune system. Like an onion, you could just keep peeling back these layers and get deeper and deeper and deeper and deeper into the immune system. This, of course, is just meant to really cover what you could see on the MCAT. So with that said, let's get into the innate immune system. So, as I said in the intro, the immune system really breaks down into two main categories. That is the innate immune system and the adaptive immune system. In general, the innate immune system is a non-specific, fast-acting branch of the immune system, whereas the adaptive immune system is a very specific system that is kind of slower to act than this innate system. And when you think of the adaptive immune system, you're most likely to think of these very specific antibodies that are produced that elicit an immune response to some very specific pathogen. That's going to be the topic of the next podcast. This podcast is going to focus on the innate immune system. So overall, the goal of the immune system is to protect our body against foreign invaders. So say, for instance, you are at a really nice restaurant and your waiter asks you, okay, how would you like your chicken done? And, you say, okay, I want it medium rare, right? You want a little bit of pink in the middle. That's typically how good chicken is. And what happens is you eat that chicken, and now you have exposed your body to salmonella, which is this little bacteria. And so now the goal of the immune system is then to kill this bacteria and prevent infection. And so, as I said before, the innate immune system is the part of your immune system that is fast-acting, non-specific, and evolutionarily speaking, it is the oldest part of the immune system. And, plant, fungi, all these different kingdoms in terms of animals, plants, fungi, all of those have this innate immune response, which points back to the fact that our long ago ancestor had this innate immune response. And real quickly, before I go on, I want to kind of clear up what I mean by non-specific. So by non-specific, I just mean that it reacts to pathogens no matter what type they are. So anything from viruses to bacteria, to some chemicals that you get exposed to, your innate immune system will react to. On the other hand, your adaptive immune system will recognize some very specific pathogen, let's say like the flu virus. It will recognize that flu virus and then say, okay, I've seen this before, I know how to respond, and then it will mount a response directly to that flu virus. So again, correlate innate immune system with a non-specific response. Alrighty, so the innate immune system is broken down into three main parts that I am going to discuss in detail. The first is anatomical barriers. The second is a general inflammatory response or general inflammation. The third is the complement system. And before I go over all of those, I want to first introduce the cells that make up the innate immune system. And I want to do this first because I refer to a lot of these different cell types when I'm talking about the different parts of the immune system. So I thought it'd be good to talk about those first so then you have some idea of what I'm referring to. So let's get into those. The most general term for a cell that is in the immune system is a white blood cell or a leukocyte. So if you hear White blood cell or leukocyte, just think, okay, this is the umbrella term for all the different types of immune cells. So then, all the cells in the innate immune system are leukocytes. And so the first group of cells I want to talk about in the innate immune system are called phagocytes. And phagocytes are cells that are capable of phagocytosing, aka eating alive pathogens. So what they do is they're able to recognize these pathogens and then kind of digest the pathogens, bring them into the cell and digest them. And so there are 4 different cells that are phagocytes. The first are called neutrophils, and these play an important role in the beginning stages of the immune response. You can kind of think of them as first responders, and typically they have these really short-lived responses. So neutrophils are the first responders, and they have a short-lived response. The second type of phagocyte is called a macrophage or a macrophage. I don't really know. I think I had one teacher pronounce it one way, another teacher pronounce it another. So I'm just going to say macrophage. And so macrophages are slower acting than neutrophils, so they're not this first responder type of cell. However, they have a much longer lived response. So they're able to kind of come in later and clean up after the neutrophils and start phagocytosing these pathogens and eating things alive. And then the next cell that is also a phagocyte in the innate immune system is called a mast cell. And mast cells play a big role in the allergic response. They release a lot of histamine, but it's also important to know that they can phagocytose gram-negative bacteria. And as I mentioned earlier with my medium rare chicken, salmonella is one of these gram-negative bacteria, and so mast cells are able to phagocytose these bacteria and get rid of them. The last cell that falls under the category of phagocyte and also is an innate immune cell is the dendritic cell or DC. And the dendritic cell is a specialized antigen presenting cell, which I'll talk more about antigen presentation in the Adaptive Immune System podcast. But just know for now that these DCs are also capable of phagocytosing pathogens. All right, so those are the 4 phagocytes that are in the innate immune system. There are also 4 granulocytes in the innate immune system. So these 4 granulocytes are basophils, eosinophils, neutrophils, and mast cells. So you're going to say, okay, wait, didn't you just talk about neutrophils and mast cells? And yes, I did, but there is some overlap in these 2 categories. So just because a cell is a phagocyte doesn't mean it can't also be a granulocyte. In order for a cell to be a granulocyte, all the cell has to do is be able to store and release granules. And typically, and especially I guess in terms of the immune system, these granules are filled with toxic chemicals, stuff like reactive oxygen species or ROS, and toxic nitrogen oxides, and some kinds of enzymes that are able to kind of chew through bacterial cell walls. So they can contain all these different type of molecules that are toxic towards different pathogens. And so what these granulocytes are able to do then, and especially basophils and eosinophils, which are kind of the poster boys of granulocytes, they're able to release these granules and direct the toxic chemicals inside of them towards these pathogens in order to kill them. So essentially what they're doing is they are shooting these pathogens with toxic chemicals like reactive oxygen species, nitric oxides, etc. So that's how granulocytes function within the innate immune system. And remember, neutrophils, mast cells, basophils, and eosinophils are the 4 cells that are granulocytes. And the very last cell that I would like to talk about in the innate immune system is called a natural killer cell. And so what these cells do is they kill infected host cells. So you could imagine, let's say somebody is infected with HIV or recently infected, and this virus is going around, replicating in these cells and then going out and infecting other cells. So what natural killer cells do are able to go to these host cells and say, okay, you, my friend, are infected with HIV, and therefore I'm going to have to sacrifice you so that you do not go and infect these other cells. And so you could say, I guess, that the natural killer cell has a passion for hunting its fellow friends. And if there was such thing as a Hunger Games of cells, it would probably be the winner. And before I dive into the innate immune system, I want to go over a few terms that you're going to hear me say a bunch of times, and that is pathogen and antigen. So a pathogen is some kind of microorganism that causes disease. That could be a bacteria, that could be a virus, that could be a fungi. Whereas an antigen is a toxin or foreign substance that induces an immune response in the body. And those definitions sound pretty similar. The difference here is that an antigen is the part of a pathogen that the body can recognize, or I guess that the immune system can recognize. So take, for instance, HIV. So HIV is a virus, it's a pathogen. However, the immune system is actually able to recognize a very specific part of HIV, and that is a protein called P24, which makes up the viral core of this virus. So therefore, the viral protein P24 is the antigen, while the pathogen is the HIV virus. So I hope that kind of makes that clear what the difference between a pathogen and an antigen is. All right, now that you have a general understanding of the cells that make up the innate immune system and some basic terms that I'm going to use quite a bit in this podcast, let's dive into the innate immune system, starting with number 1, anatomical barriers. All right, so again, the function of the immune system is to protect the body from foreign invaders. And the purpose that the anatomical barriers of the body serve is to protect the body from these invaders even getting inside of the body. So essentially the function of these anatomical barriers in the immune system is to, as Gandalf would say, tell these invaders that you shall not enter. And there's a ton of different examples of these different anatomical barriers in the body, right? There's the skin. There is the GI tract, which again is considered the outside of the body. So if you think back to physiology class, and if you haven't taken one, now you know, the GI tract is actually considered to be outside of the body. And the eyes are another one of these anatomical barriers, prevent things from getting into the body. And one that you may not think of, but is actually a very important barrier, is the blood-brain barrier. And so one thing that I think is pretty important to understand is what characteristics do all of these anatomical barriers have in common? You know, are there certain things that they all have in terms of cell type, or, you know, what makes them able to keep out these pathogens or block out these pathogens? So one of the defining characteristics of these barriers is that the cells that make up these barriers typically have tight junctions between them. So the cell-to-cell junctions between these cells are tight junctions. And I talked about what exactly tight junctions are in another podcast. I can't remember which one that was. I think it was in cell types and something like that. But anyways, a tight junction is a junction between cells that only allows the passage of very, very small molecules. In most of these junctions, most solutes cannot even pass through. So you could imagine something as big as a bacteria has a very hard time passing through these tight junctions. And it's important to note that the cell-to-cell tight junctions of the blood-brain barrier are a little bit more special. So in addition to having these tight junctions, there's another layer of security that is essentially right behind this layer of epithelial cells that form those tight junctions. And this extra layer comes from astrocyte cell projections called Glia Limitans. And what these projections look like is literally little feet. So what you could imagine is you see these two epithelial cells really close together. You know, they have this little tight junction between them that, you know, maybe if something's lucky, it can squeeze through. But then you have this astrocyte foot that's like right behind that gap that's ready to kind of catch that pathogen. So you can kind of think of it as like the game Red Rover, Red Rover. So, you know, think back to elementary school back in the good days when you were allowed to play Red Rover, Red Rover. Nowadays, they probably wouldn't allow it. But you could imagine that these epithelial tight junctions in the blood-brain barrier are like the kids that are all holding arms together and they're daring this pathogen, you know, saying, Red Rover, come on over, or whatever the phrase is. And that pathogen's going to run over. And somehow it makes it through that first layer of kids. But then all of a sudden, the teacher is standing right there. And that teacher is just like, nope, not today. And just, you know, Sparta kicks that kid right back where they came from. So the takeaway here should be that anatomical barriers are able to protect against pathogens through these tight junctions, and in the blood-brain barrier, there is an extra layer of protection that is developed by astrocyte projections called Glia Limitans. And there's another way that these anatomical barriers contribute to the innate immune system, and that is through the production of mucus. And so many of the interior anatomical barriers, such as the GI tract and respiratory tract, are covered with a layer of mucus. And what this mucus does is it protects these surfaces against microbial, mechanical, and chemical invasion. And you might be wondering, okay, what does mechanical invasion mean? And this is the classic chip sliding down the throat. You could imagine a chip turned sideways. If your throat didn't have mucus, or let's say it gets into your respiratory tract and your respiratory tract doesn't have mucus, it's really probably going to cause a lot more damage than if it did have this mucus layer, and that enables it to kind of slide up when you're coughing and prevents friction from tearing that epithelial layer. And then, maybe infections get in once that tear happens. And so it also importantly prevents pathogens from adhering to the surface of these epithelial cells. So you could imagine some kind of virus particle getting breathed in, and it's just sitting there on an epithelial cell in the respiratory tract until, for whatever reason, let's just say that this virus particle is endocytosed into the cell, and then that goes on to infect the cell. Well, what the mucus does is it essentially helps that virus particle get up into the upper respiratory tract, and then, spit out as phlegm or coughed up later on. So it just kind of helps your body ensure that these pathogens don't just sit on top of these epithelial cells for long periods of time. And so just a little digression here. There's a virus called the Respiratory Syncytial virus, or RSV, and this does exactly what I just said the virus particle in my little story did. What it does is it actually, or I guess this is my understanding of it, it is able to bind specifically to glycoproteins on the exterior of respiratory tract epithelium, and therefore it's not able to kind of get swept away by this mucus. It just kind of hangs on, and then eventually it goes on to infect these epithelial cells and cause infection in your lungs and lead to not so good of an outcome. Mucus's function in the innate immune system goes beyond this ability to kind of sweep pathogens away. So mucus is actually able to kill pathogens or inhibit their growth through these different substances that are secreted along with the mucus. And so most importantly, I guess for the MCAT, there's these molecules called defensins. And defensins are typically these short peptides that are about 12 to 50 amino acids long, and they constitute a diverse family with a broad spectrum of antimicrobial activity, which includes the ability to kill or inactivate gram-negative and gram-positive bacteria. They kill fungi, including yeast, and they also kill parasites, which are these single-celled protozoa or even nematodes, and they also kill enveloped viruses like HIV. So overall, mucus is important in the innate immune system because it's able to sweep out pathogens from these internal anatomical barriers like the GI tract and respiratory tract. And it also contains these pathogen-fighting substances like defensins. All right, so that's kind of a brief introduction into how these different barriers in the body contribute to the innate immune system. And again, that's through tight junctions and a mucus. And so the second part of the innate immune system I want to talk about, and this is probably going to take up the chunk of this podcast, is the general inflammatory response. I gleaned a lot of this information off a paper that I found on PubMed, and I'm going to attach this in the show notes, but the paper is called Inflammatory Responses and Inflammation-Associated Diseases in Organs. And the paper was by Chen and colleagues. It's a very, very well put together review. So if you're really interested in general inflammation and its role in innate immunity, I recommend going and reading it a little more thoroughly than I'm presenting it. Pretty interesting and also applicable for the MCAT. So, inflammation is a biological response of the immune system that can be triggered by a variety of different factors like damaged cells, toxic compounds, or pathogens, which is what we're most interested in this case. In general, inflammation is characterized by a few different things. That is redness of the inflamed area, swelling of the inflamed area, also heat and pain in the tissue that is inflamed. And so the redness is due to vasodilation and increased blood flow in the area of inflammation. And I'll talk about why that's important. And then the heat can come on because of a fever, and also, interestingly enough, some of the enzymes that are needed in inflammation function better at a higher temperature. And also, some of the pathogens can tend to be a little less active at higher temperatures, which doesn't make a lot of sense, but based on my reading, that's kind of what I took away. And swelling at the site of inflammation is due to the movement of immune cells and fluid into the inflamed area. Again, it will become a lot more clear why this is important as I discuss the inflammatory response. So the inflammatory response has 4 well-characterized steps that are the same no matter what triggers this inflammatory response, right? Whether that's the Ebola virus, whether that's Salmonella bacteria, or whether that's some other chemical that you've been exposed to. All these 4 steps are the same. The first step is that cell surface pattern receptors recognize detrimental stimuli. Let's go back to the case where we ate the pink chicken and we got salmonella. This salmonella would be this detrimental stimuli. The second step in the inflammatory response is that inflammatory pathways are activated. The third step is that inflammatory markers are released. And the fourth step is that inflammatory cells are recruited. So I'm going to go through these steps, break each of them down, and hopefully they become a bit more clear for you. All right, so step number 1, again, is that cell surface pattern receptors recognize detrimental stimuli. So cells in the immune system, including cells in the adaptive immune system, which I'll go over in the next podcast, contain pattern recognition receptors, which are abbreviated as PRRs. And these PRRs are encoded in germline cells, meaning that we have genes for these PRR receptors, which is kind of unlike how we make antibodies. We make antibodies as we're exposed to these different pathogens. I'll talk about this process later, but just know that that's part of the adaptive system, whereas we are born with these PRP receptors, and therefore we are able to respond to these pathogens innately or since birth. And so these pattern recognition receptors are proteins, and they are located again on the exterior of cells. And what they do is they actually bind to specific molecular patterns that are found within certain antigens or certain pathogens. And these patterns are called pathogen-associated molecular patterns, or PAMPs. And these PAMPs can include things like bacterial carbohydrates, specific nucleic acids from bacteria or viral DNA, bacterial peptides, peptidoglycan from gram-positive bacteria, and a lot of other kind of unique patterns that show up in different pathogens, whether that be bacteria, virus, or fungi.
Thanks for checking out the Prospective Doctor MCAT Basics podcast. Sam's doing a killer job taking you through the most important MCAT topics, but what if you need a little extra help? How does a 5, 10, or even 15-point increase in your score sound? Imagine how your chances at admission could increase. Med School Coach's MCAT tutoring can get you there. With the most rigorous selection process of any tutoring company, we see amazing results. We deconstruct each student, find a plan that is going to work, and help execute it. That's why our students add an average 12 points to their score. Completely physician-run and operated, and focusing on nothing but medicine. It's no wonder over 10,000 past students have trusted Med School Coach to get them through the MCAT and into medical school. Check out medschoolcoach.com today and mention code PODCAST for 5% off.
To give you a quick real-life example, let's go back to the medium rare chicken that I ordered and then I ate. I've got this salmonella that has invaded into my body. And the first thing that happens when it gets in there is that these PAMPs are going to recognize it. And what they're going to recognize is they're going to recognize a few different proteins in the flagella, specifically the FLG22 protein of the flagella of that salmonella bacteria. So it's kind of interesting, these cells are floating around and just kind of happen to bump up into this salmonella and recognize the very specific pattern on its flagella. It's pretty sweet. This example also brings up an interesting point in my opinion, and that is that this response or these PAMPs are very unique to different pathogens, right? They have to be. What if your body was recognizing like a cysteine residue or something? Well, if your immune system was recognizing just a cysteine residue, then that means that it would react to like every protein in your whole body. You'd have a massive autoimmune response and probably die pretty fast. I'm no doctor, but you'd probably die pretty fast. So it's very, very important that these pattern recognition receptors are very, very specific for pathogens or for antigens that your body actually wants to get rid of, or else you may end up having an autoimmune response. So to summarize step 1 in just a sentence, it's that pattern recognition receptors, or PRRs, recognize pathogen-associated molecular patterns, or PAMPs, which are some unique part of a pathogen, and then that is going to set off some kind of pathway intracellularly of these cells that have these pattern recognition receptors. And that is step 2 of the inflammatory response, which is that inflammatory pathways are activated. So the inflammatory pathway that's activated can be a ton of different types of pathways. These can be GPCR pathways, G-coupled protein receptors. This can be toll-like receptors. When I was looking through like a page of all the different inflammatory pathways in cells, there's just like a million of them. So, I don't think you really have to know specific pathways for the MCAT. Just know that the second step in the inflammatory response is activation of these inflammatory pathways. And I'll give you one example of an inflammatory pathway in a cell. And I'll do this, number 1, to illustrate how complex these pathways are. And number 2, to point out a few of the effects that these pathways can have on cells. So toll-like receptors, or TLRs, are a major pattern recognition receptor for immune cells, and these toll-like receptors set off a very specific intracellular pathway. And so what they first do, obviously, is they recognize some kind of pathogen or antigen. In our case, again, this is a few of the proteins from a Salmonella flagella. And so what happens is this toll-like receptor binds that protein, and as well as a co-receptor of this TLR, which is called MD2. And this triggers interactions between the cytoplasmic TIR domain of this toll-like receptor and the TIR-containing adapter proteins, which there's 3 of those. There's MAL, MYD88, and TRM. And then you have MYD88 bind another protein called IRAK4, which requires its kinase activity to then bind to the kinases IKRK1 and IRAK2, and it does that sequentially, and blah, 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 blah. You go down this huge, long pathway, and in the end, what happens is that you induce gene expression changes. And essentially what you're doing is you're increasing and decreasing expression of different genes that help the cell with a ton of different activities. That ranges from leukocyte recruitment, cell adhesion, cell survival, changes in genes in terms of remodeling the extracellular matrix, genes regulating PRRs, and the list goes on and on. So all in all, the activation of these inflammatory pathways is essentially increasing or decreasing the expression of certain genes that helps these cells respond to a particular pathogen. And more specifically, one set of genes that are upregulated are the genes that are involved in increasing inflammatory cytokines. And so this is going to lead into the next step of the inflammation pathway, which is that inflammatory markers are released. So this is step 3 of 4. So among other changes to the cell phenotypically, activation of these inflammatory pathways that I just talked about leads a cell to release inflammatory markers. And as I mentioned a minute ago, some of these markers are cytokines. And so in addition to cytokines, these cells can release inflammatory proteins, enzymes, reactive oxygen species, and more. So the first and most important type of inflammatory marker that is released by these immune cells are called inflammatory cytokines. And cytokines is just a general name for peptides or glycoproteins that regulate immune responses. So it's kind of like this really broad definition. I tried to look up a more pointed or more succinct definition, I couldn't really find anything. So this is kind of just a broad class of proteins that regulate immune responses. And so under this umbrella term of cytokines, you have interleukins, colony-stimulating factors, or CSFs, INFs, TNFs, TGFs, TGI-Fridases, all these different types of factors that essentially just help stimulate the immune response or help regulate the immune response. And so these inflammatory cytokines primarily act to recruit leukocytes to the site of infection or injury. So again, leukocytes is just a term for these immune cells. So they're just helping get immune cells to the site of infection or injury. In terms of proteins and enzymes that are released in response to the activation of an inflammatory pathway, there's a few that are important. The first is called C-reactive protein, or CRP. And I'm taking a quote from a paper here that I was reading about this, and it says that CRP binds to damaged tissue, to nuclear antigens into certain pathogenic organisms in a calcium-dependent manner. Similar to immunoglobulin G, or IgG, it activates the complement system, which I'll go into later, and binds to Fc receptors, which is part of an antibody, again, part of the adaptive immune system. So to put that all into context in more simpler terms, it binds to antigens to make them more visible to the immune system. And there's a word for compounds that do this, and that is called an opsinin, which just means that it's a compound that binds to antigens to make them more visible to the immune system. So say, for instance, we have salmonella DNA floating around, and it's so small that the immune system has trouble kind of picking it up and telling that this is a bad thing, this is an antigen. What happens then is C-reactive protein comes in, binds onto that, and makes it essentially bigger or more recognizable. So then by the time you have some other lymphocytes in there, or some other immune cells in there, it'll bind to that CRP and it'll be able to better recognize that salmonella DNA. In addition to C-reactive protein, there's another protein called haptoglobin, which clears hemoglobin from ruptured blood cells, which are called erythrocytes, out of the body. So you think about it this way: if I take a baseball bat and I just start beating the shit out of you with it, what's going to happen is you're going to break a bunch of red blood cells open, right? And that's not necessarily a good thing. It's also bad that you're getting beaten with a bat. But when you break open these red blood cells, you're going to have a bunch of hemoglobin that's floating around, in your tissues or maybe in your blood, whatever it may be. That is not a good thing. So haptoglobin is able to go in there and clear this hemoglobin and kind of start setting your body back to where it should be. The next protein that I want to talk about is called Serum Amyloid A, and it binds to the toll-like receptors to activate the immune system. So there's a ton of different proteins that are released as inflammatory markers. That's just a few of them there. In addition to cytokines and these proteins and enzymes I just talked about, you can also release different kinds of inflammatory markers. This falls under the category of other inflammatory markers. And these could be things like specific transcription factors, these compounds called isoprostanes, which are synthesized from arachidonic acid. And lastly, you can have reactive oxygen species that are released as inflammatory markers. And all these are compounds derived from molecular oxygen that usually contain some kind of free radical or some kind of charge, kind of these weird molecules that are very, very super reactive. And what's cool is there's specific cells in the immune system called granulocytes that can store granules that are filled with reactive oxygen species, and they can kind of shoot these reactive oxygen species in a way at pathogens and kill them that way. So that's kind of one of the ways that reactive oxygen species are used, and this is known as respiratory burst. All right, so that is the third step of the inflammatory response. Let's get into the fourth step now, which is that inflammatory cells are recruited. So I kind of liken this to a presidential grassroots campaign. So let's say that you like candidate X. I'm not going to name any names. And so you tell your friend about their policies and, they support this, they support that, they don't like this. And now your friend likes that candidate. And what they do is they go on and tell their friends about this candidate, and they get them to like this candidate. And it just kind of spreads in like a grassroots manner. And that's kind of a term that I always hear, grassroots. I don't know if I'm using it right, but you kind of get the point, right? One person tells another, and soon enough, all these people know about this candidate. And molecularly, what's going on is that these different inflammatory markers that have been released by other cells start to bind to and activate these different immune cells, which in turn then start to produce their own inflammatory markers, which go on and activate more cells. So you can kind of see how this response just grows exponentially like a grassroots presidential campaign. In addition to releasing other inflammatory markers, these cells are able to also say, okay, we know we're under attack by some kind of foreign substance. I'm going to go to the site of inflammation and go attack this foreign substance. So once these inflammatory markers activate these immune cells, they also start to make their way to the site of inflammation. And to me, this is where things start to get whack in an interesting way. You know, how are cells able to know where in the body to go to reach this site of inflammation? Cells don't have brains, nor do they have GPSs. They can't just plug in an address and show up at, the site of inflammation. So this process, which is called Leukocyte Extravision, is the process in which leukocytes are able to recognize the site of inflammation and get past the epithelial layer and make their way to the inflammation. And this process of leukocyte extravision has 4 main steps. The first of which is that cells that are nearby this site of inflammation start to release chemoattractants called chemokines. And what these molecules do is they stimulate the endothelial cells of the blood vessels near this site of infection or of site of inflammation to express cellular adhesion molecules. And what these cell adhesion molecules do is they prepare this layer of endothelial cells to be able to bind to leukocytes that are circulating in the systematic circulation. So now we go on to step 2, in which circulating leukocytes will bind to these cell adhesion molecules. And so there's quite a few different types of cell adhesion molecules that are expressed by endothelial cells. One category of these are called selectins. And selectins are important for a process known as rolling adhesion. Basically what happens is these immune cells that are passing by in circulation form these kind of weak bonds between selectins and carbohydrates on their surface. And so as these leukocytes kind of roll on by, they continually form and break these bonds with the selectins, and that kind of slowly slows them down while they pass along this endothelial layer. And I like to compare this to an instance I had a few weeks ago where one of the brakes on my bike actually broke, like one of the hand grippers. So what I had to do is I had to use my feet. So I was going down a hill, I realized it too late, and I was like, oh shit. And so what I did is I just kind of threw my feet down, and I would do it a little bit at a time. So I'd throw my foot down, pull it up, and that would just slow me down a little bit. Next time I'd throw it down again, slow me down a bit more, throw it down again, slow me down more, until I was stopped. So this is essentially what the selectins do. On the other hand, there are another type of cell adhesion molecules called integrins, and they bind to cells with higher affinity and cause these cells to halt. So in this case, they bind to passing leukocytes, and those leukocytes are just stuck right there on the endothelium. And in terms of biking, I liken this to throwing a metal rod into the spokes of a tire, because that just, you'd shoot off your bike, but your bike would stop right where it's at. So the takeaway here is that these chemokines cause cells of the endothelial layer near the site of inflammation to increase their production of cell adhesion molecules. I mentioned 2 of these. The first was selectins, which form these kind of weak bonds with leukocytes that are passing by and cause them to roll and slow down as they do so. There's also integrins, which bind much more strongly and cause leukocytes to stop right in their tracks. All right, so the third step now in the process known as leukocyte extravision is called transmigration. And at this step, immune cells pass through the gaps in the endothelium to get into the site of inflammation. It's pretty simple, right? So they're slowed down or stopped by these different cell adhesion molecules, and that allows them to permeate through the endothelial layer to get to the site of inflammation. And it's important to note that these leukocytes actually get two other important assists in terms of getting through the endothelium that go beyond just these cell adhesion molecules. And that is, number one, that vasodilation occurs during the inflammatory response, and this increases blood flow to the area of inflammation, which will increase the number of leukocytes, and therefore there is a higher probability of some of these getting through into the site of inflammation. And number two, the vascular permeability of the endothelial layer is increased during the inflammatory response, which makes it easier for these immune cells to pass through this layer. So that moves us to step 4 of this process, and that is that once these immune cells are able to get past the endothelial layer, they get into the interstitial fluid, and at this point, the immune cells follow the chemokine gradient in order to get to the heart of the inflammation. So this is similar to the following example. So say that I blindfold you and put you in a room. And now in that room, I put a speaker and I just start playing music from that speaker. Well, you are easily going to be able to find where that music is coming from, right? All you're doing is you're using the concentration gradient of sound. So you're saying, okay, the waves are more concentrated over here, so I'm going to go in that direction, and you head in that direction until you make it to the speaker. In the same way, these immune cells are able to say, okay, there's more chemokine over here, it must be coming from over here, and so they just follow that chemokine gradient until they get to pathogens or whatever is at this site of inflammation. All right, so those are the aspects of the general inflammatory response. Let me briefly summarize these steps. First, a cell will recognize some kind of pattern in a pathogen. Could be a pattern in the proteins of the pathogen, could be the patterns in DNA, or further things. And these are called PAMPs, and these cells contain pattern recognition receptors that can recognize these PAMPs. And then what that does is that sets off some kind of inflammatory pathway. And then setting off this pathway leads to the release of inflammatory markers, which then goes on to recruit inflammatory cells to the site of inflammation, in which these cells are eventually able to reach the site of inflammation after what is called leukocyte extravision. Alrighty, the last component of the innate immune system I want to talk about is called the complement system. All right, so I'm going to briefly talk about the complement system. It's pretty simple to understand. I'm sure it's very complex if you really want to dive in, but I'm really only going to give you a pretty surface look at it. So the complement system is an evolutionarily ancient component of the innate immune system that complements other aspects of the immune response, hence its name, and it works with both the innate immune system and the adaptive immune system. And the complement system, in essence, is made up of a bunch of proteins in the plasma that mostly target bacteria. So if you associate the complement system with anything, associate it with killing bacteria or having bactericide function. And at the most basic level, the complement system can be broken down into 3 different functions. The first of which is acting to activate inflammation. So essentially it stimulates other immune cells. We talked about the inflammation pathway. The complement system also labels pathogens for cells to recognize and then clear or destroy. And if you remember, this process is called opsonization. And all that goes on here is that proteins bind to a pathogen and make it more recognizable for the cells of the immune system. The last thing that the complement system can do in general is act in the direct killing or targeting of microbes by lysis. The classic example here is in the lysis of bacteria. And so what happens is that proteins in the complement system insert into the cell membrane to form a little hole, like literally like a Swiss cheese hole right in the cell. And then if enough of these little holes form, you eventually get more and more Swiss cheese until the cytoplasm begins to fill with extracellular fluid, and after a while, this cell just lyses. So again, the complement system acts to complement the immune system and mostly targets bacteria. So that does it for the Innate Immune System podcast. The next one that will be coming out is on the adaptive immune system, so look out for that. And stick around for a little bit of Mcat Advice of the day after I get done with this part. If you like this podcast, please go leave us a review on iTunes. It helps us with the rankings and helps us reach more students. I'd really appreciate that. As always, I really hope this podcast helped. Feel free to reach out if you have any comments, concerns, questions. Thanks for listening. All right, now for a segment I call Mcat Advice of the Day. And today's advice is pretty short and comes from my interview I had with Catherine. She's an MCAT tutor for med school coach. Our conversation covered a lot of different things, but one of the things that I thought was pretty interesting that she said was that if you don't feel ready to take the MCAT after you've studied for 3 months or 4 months, take more time. In the end, this is a huge, huge thing for getting into medical school. So she studied for 6 months and absolutely just knocked the MCAT out of the park. I was kind of under the mindset of, okay, I have 3 months to study. I'm going to take it no matter how I feel. If you've been studying and your practice tests aren't getting better, seek out some mentorship or, do something that's really going to bring your scores up to the level that you want. If you don't care about getting into the best medical schools in the nation, I say just take it when you feel like you're in a good range of getting into the medical schools you want to. If you're shooting for the top, take a year if you need it to do as well as you want on the MCAT. So put shortly, my advice is that if you need to take more time to study for the MCAT, do it, because that is the most important aspect of getting into med school, along with your GPA. Those are probably two of the top things. And obviously, it's not the only thing. Don't, extrapolate my words into saying that those are the only thing that matters, because they're not. But those are big factors. So if you need the time, take the time.
Thanks for listening to Prospective Doctor's MCAT podcast. If you're a pre-med, you'll want to check out prospectivedoctor.com for tons of free tools, articles, and more podcasts that cover your pre-med life. And if you need help on the MCAT or getting into med school,